thank the organizers for inviting me to this very interesting meet. I'm with CALVIRI, and we have a very simple mission, that is to end cancer and to do it worldwide. Now, that may seem ambitious, or most of the people in the cancer field think it's impossible, but what I'm going to try and convince you is that with a little change in your philosophy about how you approach biological problems, actually the solution to cancer is relatively simple. So most people associate things in biology as being complex. So for instance, it's thought that there's 100 to 200 different types of tumors. Each one is personal. And so the tendency is to try to develop drugs that are personal for cancer. But if you back off a little and you just forget about the biology and just pay attention to the logic of systems, you often can see very simple solutions. Let me give you an example. You probably know that every cell has a little cell within it called a mitochondria. And this is the powerhouse of a cell. And every plant has a mitochondria and a chloroplast, which does the photosynthesis. Both of them have DNA in them. They have their own chromosomes, their own genomes. And people spent years trying to figure out how to get DNA into these organelles. Very complicated and exotic experiments. Now, John Sanford and I looked at this problem and said, maybe there's a simpler solution. So what we did is we invented a gene gun. We just invented a gun that could shoot tiny little gold bullets at very high velocities. And we just shot them into the organelles. And it worked the first time we did it. And it was simple. So it's just that kind of logic that I think is more applicable to biological systems than people know. Let me give you an example that's a little harder. What about the complex problem of cancer? Now, everybody agrees that cancer is a big problem. 20 million cases, 10 million deaths. You have a 30% lifetime risk of getting cancer, and if you get it, you have a 30% chance of dying of it. It costs $1 trillion a year for the worldwide to do it. And the response of the cancer industry is basically to make more complex and expensive drugs that of limited efficacy and always have bad side effects. So what about doing something that's simpler? And I think there's a precedent for that in biology, and that's infectious disease. We have actually eradicated smallpox by making a vaccine to prevent it. And we can prevent other kinds of diseases. And the requirements to do that are really quite simple. They're that you have to find something foreign to shoot into the person. I'm not supposed to say shoot. Inject into the person and to raise an immune response to that. And you have to do it, importantly, before they're introduced to the pathogen. If you do it afterwards, it doesn't work. And so could we use this precedent that works very well in infectious disease to simply treat cancer like an infectious disease and make a preventative cancer vaccine? But to do that, you need to find something foreign in the tumors that are common between patients and across different tumor types. So it's certainly not in the DNA. So people have sequenced literally over 100,000 tumors, and all of the mutations that they have found in there that produce something foreign looking are personal. Almost none of them are shared across different tumor types. But if you think about the information that's flowing through this system, it would make sense to look downstream from this point, because as you know from information theory, there's more errors the further down you go in the process. And so what we started looking for is the next step in the process, RNA. And sure enough, RNA processing produces 100 times the number of mutations that were found in the DNA. And importantly, they're shared across tumor types and across patients. So RNA could be the source of the antigens that we need to put into a cancer vaccine, and we call them redins. But then we ran into a problem. each cancer cell can produce about 2 million different redens. And there was no technology that would allow you to search that space for the antigens that you would need for a vaccine. Fortunately, what we found is that when you have a tumor, almost immediately your B cells see it and start making antibodies to it. And they start making antibodies to these redens. So what we did is that we invented a new machine. We married the photolithography system of Intel with a peptide synthesis system and made a machine that now, instead of doing electronics on a chip, now put those 2 million redens on a chip of this wafer. So now all we had to do was take a drop of blood from somebody, put it on one of these chips, and we could wash it off and see which redens were bound by the antibody to directly read out what a tumor was making. And that allowed us to screen hundreds or thousands of people with cancer to find out what they had in common between them. So we did that, and we did that first, started in humans, but we also did it in dogs. And what we did is we screened 800 dogs with tumors, and we found out which redens they had in common and picked 20 of them to put into a vaccine. And so we created a vaccine that we predicted would prevent cancer in dogs. I'm an inventor, and I very much favor that all inventors should try their inventions on themselves. So I took the dog vaccine before we gave it to any dogs, and I can assure you it was safe, and it had minimal side effects. Woof, woof. So we did the largest dog trial ever run, 800 pets. These aren't research dogs, they're pets. We took 800 pets, screened them for cancer, showed that they didn't have cancer, then gave them our vaccine and did the trial over 5 years. And what we found, to our pleasant surprise, is that the vaccine worked. We reduced the incidence of cancer in the vaccinated dogs by 41%. Let me repeat that. It worked. Everybody said you couldn't make a vaccine to prevent cancer. You can make a vaccine to prevent cancer. And so what we're doing now is that we're creating a new version of this vaccine that we know will work much better, and we'll have it commercialized next year for dogs. And we'll initiate the formation of a human vaccine and clinical trials next year to start on the pathway to developing the vaccine for humans. So that's the point I want to make, that if you adopt, no matter what your problem is, if you adopt a simple, logical approach in biology, you can often come to very simple solutions for those problems. I'll be glad to show you other ones if you want to stop by my booth, or even try to address your problem if you'd like. But I think the important point relative to cancer, that it's possible to make a vaccine to end cancer once and for all. And we can do it. And all we have to do is have the resolution to do it. And what I'd like to have as the goal is that someday you'll have to explain to your grandchildren what cancer was. And I think that's a doable goal. And so I hope you'll support us in getting there with that. Thank you very much.